Hi guys. This week we've been asked about the mechanisms by which oxygen binds to hemoglobin and kind of the environmental and structural changes that allow this to happen within our bodies. I'll try to explain this so that perhaps someone with like a vague interest in science can get a pretty broad understanding. So, as we know, the oxygen our lungs pulls from the air is an essential molecule for cellular respiration. It enters the lungs and diffuses through the many capillary-rich alveoli branched in the lungs where gas exchange can occur. So here's an example of an alveoli taking oxygen poor blood being pumped from the heart into the capillaries of the lungs, being oxygenated, and then oxygen rich blood being basically it's going to go back into the heart and be pumped throughout the body. And then here's just a little bit more of a detailed depiction of the capillary gas exchange that occurs at the alveoli level. Gas exchange is able to occur at the capillary level in the lungs and tissue because of the differing pH levels affecting the structure of hemoglobin. So hemoglobin is the protein in our red blood cells that allows oxygen to bind to it and be carried throughout the body via the circulatory system. Here's a hemoglobin molecule right here. It's made up of 4 amino acid chains, each having a heme group, which we can see is right here. It's the little green structure right here. And then the rest of the structure is the peptide chain that the heme group is attached to. So at the center of each heme group, there's an iron ion where an O2 molecule can attach to it. Just to give you kind of an idea, there's 270 million hemoglobin molecules per red blood cell, each with 4 heme groups. And at the capillaries where the gases exchange can occur, there's capillaries all throughout our body. You can't really find a piece of tissue without a capillary only a few cell lengths away. So there's a lot. So the binding of oxygen and hemoglobin can be described as cooperative. And all that means is basically hemoglobin is an allosteric protein with identical alpha and beta subunits. And here's just a little bit more detailed picture of a hemoglobin molecule. Here's a beta subunit, and then here's an alpha subunit. And you can see here there's a little gap. You can see that this is going to be the T state which hemoglobin is going to normally exist. And basically all that means is, I guess the heme groups are these little gray points right here. So once the first oxygen molecule binds to a heme group, the structure of hemoglobin is going to change from the T state to the R state. After that first oxygen molecule binds, the hemoglobin is going to shift a little bit, and this will increase the overall affinity for oxygen. So it's going to allow oxygen to bind to the other heme groups in the hemoglobin. And then to really get into the details at the molecular level of how oxygen binds to a heme group, here we can see the structure of heme. We see the organic structure of protoporphyrin surrounding a central iron ion. And then here's like a porphyrin ring right here. So basically this iron ion is going to move into the plane of that porphyrin once the first oxygen molecule is binded to it, and it's really going to increase the affinity and the binding of the molecules together. Oxygen's affinity for hemoglobin in this heme group is actually so incredibly high that it wouldn't normally just release in the amount that we need in our tissue. So it's changed by I'm going to butcher this, bisphosphogyl phosphoglycerate, I think, 2,3-bisphosphoglycerate, which is basically going to allow the molecule to change back into the T state and allow oxygen to be released into the tissue when we need it when the particular pH level is detected, which we will talk about now. So we all know the basics of the circulatory system. Oxygen-rich blood in the arteries is sent to the tissues, while oxygen-poor blood in the veins gets sent to the heart and lungs. So as I said, the gas exchange necessary for cellular respiration occurs at the capillary level. So here is an example of the capillaries. And why this happens is hemoglobin is going to be allosterically inhibited by the acidic environment created by the waste products of cellular respiration, which we know are CO2 and water. So our various tissues and cells are working and they're expelling their waste products, which CO2 and water. So they're sitting around in the tissue, and basically those are going to diffuse into the red blood cells. And together with an enzyme called carbonic anhydrase, it's going to turn those products into carbonic acid. So the acidic environment that these products are creating, they're changing the relative pH to be higher or more acidic. So it's going to change hemoglobin's affinity for oxygen, and it's basically going to allosterically hinder what hemoglobin normally does, which is bind to oxygen. So it's going to dump oxygen, which is a really good time to dump oxygen when the CO2 and water waste products of cellular respiration are just sitting around. It basically tells the hemoglobin molecule to dump its oxygen. And then, of course, we know the waste products are expelled through our breathing. So they get taken back, the red blood cells take them back to the lungs where they can undergo the same capillary blood exchange and be expelled and then get oxygen rich blood back into the tissue. So there you have it. That is how hemoglobin is able to effectively bind to oxygen, how it's able to shift its structure in a way that allows oxygen to completely saturate the heme molecules, and how it's able to effectively release oxygen into the bloodstream when we need it, when the acidic environment is detected as it is caused by cellular respiration, the byproducts of cellular respiration.